Okay, good afternoon, everyone, and welcome to the event. We'll get started in just a moment, but before we do, I just wanted to give a few logistical remarks. We are recording the event, and the video will be available for you to view on the Basic Science website. You and others can view the video from this event and others from this season and past seasons at the Basic Science website. We'll post that link in the chat. Due to the large number of people that we're expecting, we are going to keep the audience muted throughout the event, but we do want to hear from you. So if you have any questions for our panelists or comments, please post them in the chat. We'll be monitoring the chat for your questions, and we'll try to get to as many of those as we can. And with that, it is my privilege to introduce the Dean of Biological Sciences, Mike Bateup.
Good evening, and welcome to Basic Science Lights the Way. As you've heard, I'm Mike Bateup, the Dean of Biological Sciences and also a professor of molecular and cell biology. And I'm really so pleased to see that you're all here in such great numbers for another series of these talks. I find the topic, this particular topic, so interesting and exciting. It is a great inspiration to end my busy Wednesday. And of course, this is going to feature one of Berkeley's great traditions and great strengths, and that is a basic science research. And the goal of this series in general is to shine a light on how scientific discoveries are made and what it means to be driven by curiosity. Basic doesn't mean simple, but rather foundational at the core of things, in that it can lead to transformational discoveries. At Berkeley, we often point to the well-known examples of discoveries that were made because our scientists followed their curiosity. This curiosity has led to translation, including immunotherapies for cancer, the CRISPR-Cas9 gene editing, the accelerating expansion of the universe, and the first reconstruction and visualization of brain waves. While these breakthrough achievements are certainly worth celebrating, none would have been possible without a solid foundation in basic sciences. Tonight's theme, as you know, is the life of a brain from development to degeneration. It's a serious topic for us all that we hope will give you a glimpse into the lifespan of this marvelous organism. Our moderator this evening is Dan Feldman, a professor of neurobiology in the Department of Molecular and Cell Biology, and he's a member of the Helen Wills Neuroscience Institute. Dan's interests span neurobiology, learning, neurophysiology, and sensory biology. Dan's lab studies how neural circuits in the brain's cerebral cortex mediates sensation, memory, and higher function, and how this breaks down in neurological disease. Once we get underway, I just want to reiterate what you've already heard. We encourage you to post your questions for any of our speakers in the chat box. We'll answer as many as we can. While we can't possibly be comprehensive in an hour, and it's always too short, we hope to inspire your curiosity to learn more at your leisure. Okay, Dan, it's your show now.
All right, thanks, Mike. Welcome, everybody. We're here tonight to talk about the brain, and I'm very happy to be moderating this event, and I'm really pleased to actually be able to announce to you all that Berkeley is working to establish a new department of neuroscience. This would be a new department on campus. The work of this department will be centered on understanding the brain from molecules to mind, from cells to computation, and from science to society, and we're really excited about this prospect. I'm sure one reason you're all here is that you know that how the brain generates behavior and perception and cognition and our internal mental lives is one of the really great unsolved challenges in biology. And it's important not just for answering these fundamental questions, but also for understanding what goes wrong in the brain with disease, which of course is critical for individuals and for society. And this is not just a biological problem, because the brain, one of its remarkable aspects is that it's a computer. It's a biological computer, different from your desktop or laptop computer, a computer built of fat and salt and protein. And how it actually computes is a fundamental mystery. And to solve that mystery, we need to bring together biologists, computer scientists, engineers, chemists, psychologists to really address this problem from all sides. And this is what the department will do. So we'll do lots of exciting research, make lots of great discoveries. We're going to provide a new comprehensive neuroscience major for undergraduates who want to study the brain. And this will really consolidate Berkeley's role as a national leader in neuroscience. So I'm very glad I can share this with you. So it's a delight for me to participate in tonight's event. And I'm excited to introduce you all to some of my colleagues and their exciting work. We are going to hear from three inspiring faculty members, Professor Helen Bateup, Michael Yartsev, and Daniela Kaufer, and they will talk about some of these deep puzzles of the brain and the basic research that illuminates it. At the end of the talk, we'll try to address all of your questions, so please put them in the chat. So our first speaker is Professor Helen Bateup. I'm going to stop sharing my slide here, and while she shares hers, I will introduce her. Professor Bateup is an associate professor of neurobiology in the molecular and cell biology department, the MCB department. Her research focuses on understanding molecular and cellular neuroscience, brain development, and neurodevelopmental disorders. And she'll be talking today about human brain development and how it can be studied using human brain organoids grown in a culture dish in the lab. So Helen, can you tell us about your research?
Sure, thank you so much, Dan. Hi, everybody. It's a pleasure to be here. Thank you for joining us on this discussion about the brain. So as a neuroscientist, I've been fascinated by the process of how an organ as sophisticated and complex as the human brain develops and functions. So on this slide, in the schematic, you can see how the brain goes from a very simple neural tube and turns into this highly elaborate and complex structure over the course of pre- and postnatal development. And if we look at what's happening at the cellular level, we see a number of processes that need to be properly coordinated in order to produce this highly elaborate structure. And before birth, around 86 billion brain cells are born. These brain cells are called neurons. After these brain cells are born, we have another 85 billion non-neuronal cells, which are glia and astrocytes and other support cells, which need to be born. And these cells form coordinated networks and connections.
Hello?
Hello? Okay, sorry about that. These cells need to wire together and form these connections, and these brain cells communicate with each other via synapses. And this development of these connections and these synapses happens early in prenatal life, but also during early postnatal life. And I want to highlight that here. So here's an example schematic of, these are cells, individual brain cells in the cortex of a newborn human. And you can see these cells are born and they have these rudimentary processes. And over these first few months and years of life, these neurons grow and develop these dendrites, which will form connections with other brain cells. And this process continues through childhood. And then at some point, we can see the refinement of these connections in a process called experience-dependent learning. So this is a very dynamic period in terms of brain development and function. And my lab is very interested in understanding the molecular processes that allow this dramatic development to occur. And of course, we know that this process requires really precise coordination at the molecular and cellular level. And when things go wrong, we can see the emergence of diseases, especially neurodevelopmental disorders. So these disorders can include things like intellectual disability or developmental delay, childhood onset epilepsy, and other behavioral and psychiatric conditions like autism. And so my lab is also interested in understanding when these processes, developmental processes, go awry. How does this give rise to brain disorders? And so, of course, we don't have access to the human brain to study its development at the molecular and cellular levels, so we require model systems in order to study these processes. And you're going to hear about some of these animal models that have been really important for our basic neuroscience understanding of brain development and function. But I just want to highlight that the human brain really is unique and special. And so here's a cross-section of the human brain. And in comparison, you can see a rodent brain, a mouse brain. And the human brain is really dramatically expanded over evolution, especially if we look at this outer structure, which is the cerebral cortex, that has really evolved considerably over time. And this brain region, the cortex, is what's thought to give us our extraordinary cognitive abilities. And we know that the development of the mammalian brain is very similar between mice and humans. However, there are some special cell types that are only present in the developing human brain. And it's possible that some of these neurodevelopmental disorders could arise from dysfunction in these very human-specific cell types. So to overcome this challenge of how do we model and study the human brain, we and others have developed a pipeline that allows us to actually grow a miniature model of the human brain in a dish. And for this, we capitalize on the expertise of our colleague at UC Berkeley, Dirk Hockemeyer, who's an expert in human stem cells. And what we can do, through advances in this stem cell and gene engineering, we can now take skin cells from a person or a patient with a disease. We can so-called reprogram these cells into pluripotent stem cells in a dish. And these human pluripotent stem cells have the ability to give rise to, in theory, any cell type in the body. For our interests, we can supply a cocktail of different factors that are normally present during the developing brain, and we can cause these stem cells to differentiate or transition into neural progenitor cells, as well as different types of neurons. And more recently, we have the ability to develop these three-dimensional brain organoid structures that really model the very early developmental processes of the human brain. And I also want to add that we now have the ability to use tools like CRISPR-Cas9 that were developed here at Berkeley to perform gene editing of these stem cells in order to introduce mutations or to even repair patient mutations and study how they affect brain development. So in the next couple of slides, I'll just show you an example of some of our research that has made use of these brain organoid models to understand human brain development and what goes wrong in disease. So here are some images of brain organoids as they develop in the cell culture dish. And they start out as mainly neural progenitor cells that divide and grow. They transition to producing neurons or brain cells, and later on produce these support cells like glia. And this process mimics early human brain development. And here you can see one of our brain organoids where we have made a cross-section, and we can see these developing cells, and they form these rosettes of neural progenitor cells that divide and give rise to newborn neurons. And we can watch this process happen in a dish. And one of the amazing things we can do is we can grow these brain organoids for very long periods of time. So here's a 1-year-old brain organoid that we have in the lab. And these will continue to develop and mature over time on roughly a human timescale. And so, as I said, we've used this new model to understand really the cellular, molecular, genetic basis for normal human brain development. We've also applied this model to understand disease. And so one of the diseases we've been particularly interested in the lab is this neurodevelopmental disorder called tuberous sclerosis complex, or TSC. And this is a genetic disorder. Patients with mutations in the TSC genes present with an array of manifestations, and the most prominent of which is the presence of these cortical malformations, which are these regions of abnormal and dysmorphic cells that form these clusters that you can see on an MRI. And these arise during embryonic brain development, and these cortical malformations often become seizure foci and will cause epilepsy and other problems in individuals with TSC. And because these tubers arise developmentally, it's really been an open question in the field where they come from, and we need a model to study them. Here is an example of what the cells look like in one of these cortical malformation regions. And this is tissue that we had obtained through a collaboration with Stanford University, where these tubers were surgically resected to control epilepsy in a child with TSC. And you can see these really abnormal looking cells that are neurons and these support cells called astrocytes, and these really strange cells that don't normally exist in the brain. And we really wanted a model system that captured this pathology so that we can understand where these cells come from and how they cause pathology. So we made genetically engineered human stem cells that carried these same mutations that the patients have. We differentiated these stem cells into these brain organoids. And what you can see here is a section from about a 4-month-old brain organoid, where in red is labeled the cells that have this mutation that occurs in people. And what I hope you can see is that we can recapitulate these regions of abnormal and dysmorphic cells within the brain organoid that really resemble these so-called tubers that we see in patients with the disease. And again, if we zoom in on what some of these cells look like, here's an example of these abnormal and dysmorphic cells in a patient sample. And here are some of the cells that we see in our brain organoid model. And so they look very similar. So we really think that we have a new way to capture this pathology of the disease. And this was the first human brain organoid model for this disease that we published together with the Huckemeyer lab in Nature Medicine. And so we've taken advantage of this model to interrogate the molecular mechanisms that cause the abnormal development of these cells. And right now we're pursuing strategies to understand how these abnormal cells cause dysfunction and abnormalities in the surrounding cortex, and how this might give rise to seizures and other problems that individuals with TSC face. So with that, I would just like to summarize by saying that human brains are amazing, they're large and complex. And to establish such a complex organ, we need really precise coordination of genetic, molecular, cellular programs that occur both during prenatal and early postnatal development. And human brains in particular, due to their complexity, develop over a protracted period of time, which can allow for vulnerability, and really alterations in these developmental programs can lead to neurodevelopmental disorders. And what I've shown you, or what I've highlighted today, is that we have now the ability to use human stem cell derived brain organoids as a new model to investigate normal human brain development, as well as the processes that go awry in disease. And so I'll end here and just show the people in my lab who do this work, the students and postdocs, as well as our many collaborators at Berkeley and elsewhere. And I look forward to taking your questions. Thanks again.
All right, thanks, Helen. That was terrifically exciting. I find it really amazing that you can grow these little human brain organoids and watch them progress in their normal development, you know, and study what goes wrong in development. And actually, there's a question that I wanted to ask about that, which is, you say that these organoids develop in a dish at roughly a human timescale, but of course they're growing, you know, outside of their normal environment. So what sets that normal timescale for development? Is there some clock in all of our cells that's making humans develop slowly, but little animals develop more quickly? Is it just that we're bigger? What's going on?
Yeah, that's a fascinating question. This is one of the most intriguing things to me about working with the brain organoids, is that early on in the process of generating them, in the first couple of days or weeks, we supply factors that encourage them along to differentiate along a neural path. But after this initial patterning, we don't do anything to the organoids, we just let them grow. And so they spontaneously mature and develop over time. And so it really is, you know, in the absence of any other cues, it really is an intrinsic developmental program that these cells follow. And one of the most fascinating things that has come out of the brain organoid field recently is that it's known that their cells have different signatures in the fetal brain than in the postnatal brain. And we can see that in the patterns of genes they express. And these brain organoids will spontaneously switch around 9 months in culture from having a prenatal or fetal signature of gene expression to a postnatal signature of gene expression. Again, this is all happening in a dish through some spontaneous intrinsic timing. And so I think that's really fascinating that we can capture those developmental trajectories in this model.
That's great. There's a related question to that in the chat from Tanya. She asks, what part does electricity or electrical signaling play in the development of these neurons?
Great question. So we can see the emergence of patterns of neural activity in the brain organoids. Again, they are representing a development of the brain. And so it takes a few months of growing these organoids for this activity to emerge, for the neurons to be mature enough to allow them to form synapses and fire action potentials. But we can see patterns of activity within these organoids. And there have been studies showing that in models of neurodevelopmental disorders, we can see abnormal patterns of activity in these organoids, which resemble abnormal brain activity in patients. So this is a very new area, and characterizing this electrical activity and this synaptic function in these organoids is really a new and emerging area, but we think that we can do that.
Great. So then Diane wants to know, are there other forms of autism which seem to also involve unique or different cell types, or is TSC unique in that?
Yeah, so in our lab, we have worked a lot on TSC, but we're also very interested in other autism spectrum disorders. And so we're looking at other genes that are known to confer risk for ASD. And I think that is a key question, is defining which are the cell types that are really responsible for driving these manifestations. And we have looked at the cortex, of course, but we're also exploring other cell types. We think that there are some shared features across different types of autism spectrum disorders that we can see in these different model systems.
Okay, I'm going to combine a couple of questions together because they're related. Thanks everyone for submitting the questions. This is so exciting. Let's see. We have a question from Kevin. Can you artificially speed up aging to study diseases like dementia? And that's related to a question of, is there some limit on the amount of time or number of cell cycles that you can replicate, cells in the dish?
Yeah, great question. So of course, it would be great if we could use these brain organoids to model neurodegeneration, because this is as we'll hear from Daniela, the aging brain and neurodegenerative disorders are a major challenge for society. As I said, these really are, organoids are reflecting kind of earlier or younger human brain tissue. And so we do need to think of ways about how we could model aging in this system. And there are some tricks that we can do to artificially try to age the cells. And that's something that people are employing. And I'm sorry, I forget the second part of that question.
Is there some limit to how long you
Okay.
can grow them now?
Yeah, I mean, we have grown these brain organoids for a year or more. There are studies published where the organoids have been grown for up to 2 years. There is a limit on how big they can get. So they grow to be about 1 to 2 millimeters. And once the progenitor cells have all differentiated, all turned into neurons, they don't grow or divide anymore. But those cells that are in the organoids do continue to mature. And yeah, I don't know what the oldest brain organoid is, but certainly a year or 2 is possible.
Okay, so then I have to ask the broad question of how similar are these organoids to a real human brain? And if you let them grow, to their maximal size, do they become little thinking human brains in the dish?
Yes, so that's an important question. We have to think about some of the ethical issues surrounding this. So in early iterations of this approach, people would nickname them mini brains, or, in the media, they would be called mini brains, but I think that is no longer favored because they really aren't mini brains. They're really, organoids or spheroids, which is a ball of cells. And they reflect some features of the human brain, but they certainly are not mini brains. So they aren't lacking all the cell types that you would normally see in the human brain. The cell types are not exactly in the same proportions. They don't have blood vessels. So they are the best approximation that we have of the human brain, but they certainly have a long way to go before they would be considered an actual functioning brain.
Okay, thanks, Helen. That was really terrific. Thanks for that great talk.
Thank you.
So we need to move on to our second speaker. So I'm happy to announce that our second speaker is Professor Michael Yartsev. Michael is an assistant professor in the Department of Bioengineering and also in the Helen Wills Neuroscience Institute. And his work revolves around complex questions about how the brain processes information for behavior, particularly complex behaviors like navigation and communication and social behavior. And he does this by studying a remarkable mammal, the bat. So tonight, Michael will be talking about bats as a model system for understanding spatial navigation and spatial memory. Michael, take it away.
Okay, thank you very much for the introduction. And thank you all very much for joining. It's really an honor and a privilege to be able to share our research with the community. So as Dan mentioned, we are interested in the brain mechanisms of spatial navigation. And today I'm going to tell you about some of the work my lab has been doing on how space and time and our memories are represented. And we utilize bats for this particular type of questions, and I'll explain why. So I'll mainly focus today about the brain's GPS system, kind of an internal sense of our knowledge of where we are in the environment, and a particular structure, as noted by Dan earlier, that is very clinically relevant, known as the hippocampus. This is a structure, a key structure that gets damaged during diseases like Alzheimer's and so on, where basically people, humans, and also in the case of animals, lose a sensation of space and our ability to form new memories of our environment, including our homes, neighborhoods, and so on. And within that structure, there's a particular type of cell known as place cells. And instead of showing you or telling you about it, I actually want to show you in real life how such a cell operates. So this is a movie from the Moser Lab in Trondheim. And what you're going to see and hear is how does that cell operate. So this is a recording from the hippocampus of a rat as it moves around the environment. The crackling noise you just heard is the activity of this particular one neuron in the hippocampus. And hopefully what you see as the animal kind of moves around the environment is that this neuron activates in a particular location. You can try to guess where that location is, but basically this is believed to be one of the mechanisms by which we get this sensation of space, this GPS knowledge of where we are in the world. So you can see the animal goes again and the neuron fires. So this is the kind of maps we get. And when recording from the hippocampus, this work was also awarded the Nobel Prize in Physiology or Medicine in 2014. And here you can see the trajectory of this rat as it was moving around. The red is the activity of this cell, and here below is a color-coded image showing this cell was active in the top right corner of the environment. Now, similar responses have also been shown in humans, so we know this is a very human-relevant phenomena. And also in earlier studies in bats I've done, we found these place cells also to exist in the bat when it was crawling around in 2D. But now really, let's get back to the question of why are bats such a useful model system for addressing these questions about spatial cognition and memory? So there's multiple reasons, and I'll mention some of those as we go through the talk today. So the first one is that the world is not flat, but actually 3D. And bats are very advantageous for studying three-dimensional space because they are the only flying mammal on the planet, right? So they naturally move and navigate in 3D, which makes them ideal for studying how is 3D space represented in the brain. So as Dan noted, half of my life is engineering. So at the time we developed these tools to wirelessly record from the bats as they were flying around, which was not possible at the time. And this allowed us to understand how is 3D space being represented by those neurons. So this is an example of one such cell. You can see it basically looks like a ball, suggesting that 3D space was volumetrically represented, and all the neuron really cared about all the different axes of the environment. So the X, Y, and Z kind of axis of the environment. And this was really, really interesting. But more recently, kind of also as noted by Mike, here in Berkeley, we're very much driven by curiosity. And we were curious is this enough? Is it enough for us to just know where we are in space at this present moment? Or do we also need to know where we were in the past and potentially where we're going to be in the future? And you can think of it in a very intuitive way. As you drive around in your car, approaching an intersection, it's really useful to know where you are right now, but you would really want to know where you're going to be half a second from now, right? Kind of project yourself to the future, position-wise, to avoid a potential collision. So why are bats such a useful model system for that question as well? Because unlike a terrestrial rodent you will typically find in a laboratory, bats fly really, really fast. Actually, they fly the speed of the same speed that you would be driving a car, about 40 to 50 kilometers per hour. So they're ideally suited at looking at neural activity, which is happening over a very short temporal time, short amount of time, during which the animal, much like you driving a car, can move a very large distance. And to make a rather long story short, we found that actually most of the activity within the hippocampus is really not just about where you are right now, but it's about projecting yourself into the future predominantly. And you can see an example of one such neuron here. So this is a 3D map like the one I showed you before. In this particular example, you can see the activity of this neuron is rather mushy, right? Kind of happening all over the place. These are red dots. But if you now take this activity and relate it to where the animal will be just over half a second from now, it becomes incredibly precise. And that's actually what we found for the vast majority of neurons in that structure. So this was very interesting, but then we wanted to go one level up, right? So when we think of hippocampus and Alzheimer, the first thing that springs into our minds is memory, right? Our memories of our life, of our childhood, everyday life, memories lasting days, weeks, and months, whereas here I was talking about seconds at most. So how are our long-term memories represented in the brain? And just to kind of put things in context, I bet all of you can now remember, most of you can remember your way back to your childhood home, right? And even if you go with your memory to your childhood home, you can remember, how does that house look like, right? How did you navigate between different rooms? How do those rooms and your experience within those occurred? But we don't fully understand how does the brain represent this long-term memory of space. So again, why are bats such a useful model system, right? And the reason is because our particular species, the Egyptian food bats, are amongst the world champions of long-term spatial memory. So I'm just giving you a snapshot of about 3 months in the life of a bat. This is data from the lab of Yossi Yovel in Tel Aviv. And what they do is they track the position of those animals as they navigate outdoors. And here different lines represent different navigation trajectories a given bat took. And every line is one single night. And this is the scales of tens of kilometers. And hopefully what you can see is these animals can navigate with these extremely precise trajectories to very distal home reward location, if you will, suggesting they really have this capacity of retaining a very long-term memory of very complicated environment. Now, fortunately for us, also within the laboratory, these bats will navigate in this extremely precise way. So you're seeing here a top view of trajectories taken by this bat. Different colors, a different trajectory, and I'm just showing you all the trajectories this bat has taken over many days and weeks, and hopefully you can see how precise those are. And this is a huge advantage over what we could do up until now in neuroscience, because now we can ask, how does the brain represent not only the same environment, but also similar movements within the environment? Like imagine you navigating from the living room to your bedroom or from your home to work, following the same trajectories. How are those long-term memories actually represented? So the behavioral side, this is a big advantage, but what about the neurotechnology needed to address this question? So again, putting kind of on my engineering hat, we had to establish the tools that will allow you to monitor the same brain activity over the same prolonged periods of time. This is done using tiny miniature microscopes that can be placed on the head of the animal that's flying around, and you can literally visualize in real time how does the brain operate. So what you're seeing here, those flashing cells, are these hippocampal neurons active in real time as the animal navigates, and importantly, it allows us to observe the same cells over many days, weeks, and potentially maybe even months. So what's the end result of all of this? What's the solution? The solution was that it seems that the code within our brain for space is actually incredibly stable. And you're seeing an example, one neuron here, and I'll show you all the data in just a moment. So this is a color-coded image over many, many iterations here, 100, 200 trials. The bat was flying the trajectories over multiple days, and hopefully you can see the code remains exactly the same. The neuron fires in exactly the same location day in and day out. And if we now take all the hundreds of cells we monitored and align them relative to day one and look at them across many days, and we even looked for longer, but here showing you just for 5 days, you basically see the code remains exactly the same. So to summarize, what I hope I was able to show you is that our GPS system in the brain, our positional system in the brain, is actually incredibly stable. And it is largely non-local, so it is located in time, and it extends together along the full continuum of both space and time, and giving us this sense of spatial memory and ability to navigate, which we all have. So I want to conclude and, of course, acknowledge all the wonderful people in the lab doing the research, our funding sources, and thank you all very much for paying attention.
All right, Michael, that's terrific. Thank you. So bats have incredible, long-lasting, stable memories of spatial environments and how to navigate. That's really exciting. And I thought it was also a nice example of how at Berkeley, one of the things we do in research is build the technologies in order to make the discoveries for how the system works. And clearly your lab does that. I have a question for you to start, actually, which is that are there any lessons that you can take from your work based on how the brain is processing these types of navigation, information, and memories to design navigational systems or artificial intelligence systems that might be useful to people?
That's an excellent question. Thank you, Dan. So indeed, I believe there's like two main goals, largely why we do this sort of research. The first one is to help understanding of the human brain, solve diseases, and so on. That's obviously very important. There's also a basic science approach, which is the principles we learn, as you noted earlier on, the brain is a computer. The principles we learn here can be implemented, right, in a variety of ways. And indeed, the lessons we're learning here about how does the brain compute and allow complicated forms of navigation can be very easily applied, for example, in self-driving autonomous vehicles, which is basically a navigation problem, right? Like a Tesla needs to get you from point A to point B. That's the main job of this car. And similarly, you can take the principles like how you dislocate in time, how do you compute and retain a complicated representation of your environment and implement it there. So that's something we're actively pursuing right now. And I very much hope, so if you see like a little Batmobile driving around the Bay Area, we had maybe something to do with it.
Yeah, and I mean, for people who don't think about how the relationship between neuroscience and other forms of engineering and development, I would point out that what we call neural networks, which are a brain-inspired way of designing computation, and these are the guts that underlie computational engines we use all the time, like in Google Images and Google Translate and in Maps, for example. They really came very directly from discoveries of how neurons talk to each other and store information dating back to the 1980s, in some cases 1970s. And so there's really a development in both ways between neuroscience and engineering and computer science as well. Okay, some questions from the chat. So is it really true that if I'm walking around my house, my place cells are firing?
Supposedly, yes. So in humans, it's very difficult to do, right? So in humans, these recordings were done primarily in the operating room. So for example, one of the other important functions of the hippocampus, like this function, I should say, is a lot of seizures originate within the hippocampus. So a lot of time people who reach the OR kind of in an epileptic state where a neurosurgeon will need to identify the locus of the seizures, and typically it will be in the hippocampus. So in these rare cases, you can record from the human hippocampus, and it's hard to get a human move around in the OR, so they do it in virtual environments. So kind of imagine yourself navigating in your virtual home, and indeed, in these cases, if you monitor the brain cells in the hippocampus of a human, you find neurons to be very spatially selective. So seemingly that, yes, that this is a very conserved principle existing all the way from very simple mammals all the way up to humans, which, it's quite interesting and impressive.
So there's another question from Hari. Hari wants to know, does the brain's representation of where an animal is in space also relate somehow to its understanding of its own body layout and how its body is oriented?
Well, that's a very good question. So you can think of space as being represented in different coordinate systems, right? So you can have like an allocentric and an egocentric representations. And so generally the way we think of hippocampal representation is how do I relate to the environment around me, to objects around me, and the layout of the environment around me. For example, the window is on the right, the door is on my left, and I need to go straight between them to reach the kitchen. So that's more like hippocampal representation. There's certainly other structures in the brain that are believed to more involved in how do you orient yourself like physically. For example, I'm now moving my head 50 degrees to the left relative to my body coordinates. So there's different types of coordinate system represented within our brain in different structures. So the hippocampus is much more of a relational map of me as a figure relative to my environment.
Okay, thank you, Michael. That's great. There's a lot of other great questions, but we're running short on time, so we need to move forward to our third talk.
Absolutely, thank you.
So thanks, Michael. So the third talk tonight is going to be given by Professor Daniela Kaufer. Daniela is a professor in the Department of Integrative Biology and also in the Helen Wills Neuroscience Institute. And her research spans many areas from neuroscience to aging, to dementia, to stress, to neural stem cells, brain development, traumatic brain injury. But today she's going to be presenting on her discovery of important new mechanisms for aging in the brain. Daniela, tell us about your work.
Great. Hi, everyone. Thanks, Dan. Thank you all for joining us. And we're now getting to the exciting topic of degeneration and disease. So my lab had been studying variability between individuals. And we're looking specifically at the variability that is seen when one is faced with psychological trauma or stress or brain injury. Some individuals are resilient and will show no long-term clinical effects after the incident, and some individuals are vulnerable to developing long-term neurological or psychiatric consequences. And we've been working on understanding the active mechanisms that underlie both of those sides, vulnerability, but also resilience. A lot to learn from each of those. And you'd be surprised how little is known, and even less is known about aging, natural aging, not just in the context of neurodegeneration like Alzheimer's disease. And aging is similar because although there's no defining incident, if we follow trajectories, some individuals age very successfully and others are vulnerable, and they will show similar neurological and psychiatric symptoms as you would see under injury. And we asked ourselves a question trying to understand something about the consequences of those two events, comparing those and seeing if we can get something of mechanism here, asking if there are conserved regulatory mechanisms that control some stereotypical first steps in progression from a healthy brain to an aged brain or to an injured brain. And the answer that we found by comparing those two scenarios was actually a very surprising one, and it gave us a new framework to think about mechanisms of dementia and cognitive function. Mostly when we think about brain function and brain diseases, and the two talks that you've just heard now, the focus is on neurons and those cells that are the basis of the electrical signals in the brain. What we discovered in this project that I'll show you today is that the quality of the brain environment is actually a critical component in the development of pathological processes. So what do I mean by brain environment? The brain blood supply is a system of blood vessels that's responsible to supply massive glucose and oxygen demands of the brain. And this system, the vasculature in the brain, looks a little bit different than the ones that we find all over our body, because it's meant to keep the brain environment very much protected. It doesn't want toxins and pathogens and cells and molecules that are in the blood to gain access into the brain. And so there's a filtration system that exists in those brain vessels that keeps the brain protected. And recent technological development allowed us and allowed other labs to create, using accompanying algorithms, maps that quantify blood-brain barrier dysfunction. How would you do that? We do that using what we call dynamic contrast enhanced MRI. This is an MRI technique. There's a magnet that scans the brain while we inject a tracer IV. In the case of a healthy, intact blood-brain barrier that works really well, we will not see the tracer in the brain. So you can see this control individual here, for example, or here. We're not seeing any of the tracer. But if there is some blood-brain barrier dysfunction, we will see that area of the brain light up with those colors, and the warmer the colors, the more tracer that is there. And this method was used to document chronic situations where there's blood-brain barrier dysfunction. So you see here some examples: chronic epilepsies, Alzheimer's disease, Parkinson's disease, ALS, bipolar disorder, currently very relevant COVID, are chronic situations where we see blood-brain barrier dysfunction. In collaboration with Alon Friedman in Ben Gurion University in Dalhousie in Canada, we've used this method of DCE MRI to try and quantify blood-brain barrier function in this injury and aging scenario and ask questions about what happens then. And so what we're looking at here is blood-brain barrier function in college students that are players of American football, and they're compared to control subjects on the track and field team. So those are the players, the team members on the track and field team, and you see that we're not seeing, they're young, healthy individuals, we're not seeing a lot of tracer in their brains, but when we look at the football players, in a third of them, we found some blood-brain barrier dysfunction at the end of the season. We also, I'm not showing you here, but we've developed in the lab an animal model to look at that, and we know that we're seeing vulnerable individuals with blood-brain barrier dysfunction, and those are also the ones that are prone to long-term neurological complications. So we then took those techniques and applied them to natural aging. We looked at healthy individuals from 20 years old to 80 years old, and we looked at mice that we naturally aged in the lab. We let them be born in the lab and then live their whole life, about 2 years of age, in the lab, and we're looking at their brains, and what we could see is that there's an age-dependent blood-brain barrier dysfunction. We see that in the older individuals, mice, in human, we see this color of the tracer, or we see a red dot here that is a blood protein that entered the brain. Where in the brain? The area that you just heard about, the hippocampus, this area that is important for learning and memory and navigation, is the area where we see this blood-brain barrier dysfunction. And if we look at the numbers, we can see that it starts somewhere around middle age for some individuals, and when we get to people that are over 70 years of age, about 60% of them, of us, will show blood-brain barrier dysfunction. So we then used the next, 10 years or so to try to understand what happens in the brain when the blood-brain barrier is leaky. And there's a lot of steps here that we found, so I can't really tell you all of them, but I'll tell you that we have now a step-by-step understanding of what happens when the brain now sees proteins that it wouldn't otherwise. Those are proteins from the blood that get access into the brain. They act on specific receptors, pro-inflammatory receptors, receptors that promote inflammation in the brain in those other cells, in those astrocytes, the cells that Helen told you come later. There's the support cells. And what happens now is that there is such a massive inflammation as if there was actually a foreign pathogen in the brain. And this massive inflammation changes the function of those astrocytes, and then it changes the function of the neurons around them, and it changes neural activity in a way that we predicted should show abnormal synchronized activity. And so to check that, we actually put little electrodes like EEG, but this time inside the brain in mice, and we recorded them. And what we were able to find, to discover, to identify is a signature of indeed synchronization. And this signature shows this little bit of a slowing of the brain. It occurs through a few seconds. You might think about it as the spinning wheel of death that you might see on your computer when it's sort of bogged down and slows down. But we see thousands of those events per day in a particular group of vulnerable individuals. So young animals do not show that when they're awake. Old normal animals hardly show that, but then those pathological vulnerable population shows thousands of those events per day. And we see them in aged mice, we see them in a model of Alzheimer's disease, we see them in Alzheimer's disease patients and in dementia patients. So this is a beautiful correlation that I'm telling you about, but that does not really shows us, that does not definitively tell us that blood-brain barrier dysfunction is what causes the neural dysfunction in dementia. So to actually ask this question, we went about it two ways. We wanted to see whether we could do a gain of function. We take a young mouse, intact, healthy, and all we're going to do is mimic blood-brain barrier dysfunction in it. We're going to put a little pump in it and take this blood protein and infuse that into the hippocampus and see whether this is enough to recapitulate aging in that mouse. And on the other hand, we were even more ambitious and we said, now let's look at loss of function. If I were to fix this blood-brain barrier, can I fix aging, brain aging? And we did that in two different ways. Genetically, we knocked down TGF-beta receptor, this protein that detects the blood protein in the astrocytes, or pharmacologically, because this is more relevant if we want to think about drug development, we developed a small molecule inhibitor of that receptor that fixes, that restores the blood-brain barrier dysfunction. So let's look at what happened there. Let's start with a gain of function. We take a young mouse that is healthy and well, we put this little pump in, and after a few days or a week of infusion of that blood protein, we start seeing signatures of aging. We see gene expression that mimics the gene expression in the aged brain. We see synaptic transmission, inflammation, senescence of cells as we would in an aged brain. When we record those animals, we can record animals that have a control pump that only pumps saline, or a pump that pumps this blood protein. And we see that we start getting, again, thousands of those events per day in those animals. Now, what would be an important hallmark is what happens cognitively. And so what we did is we tested those animals in a water maze. We teach them how to learn, or we let them learn how to find a little platform in a water pool. They don't like swimming, and so they try to find this water pool. And if it's a young mouse, within 5 days to a week, they will very quickly learn the way and go directly to that platform. An animal that have a pump that have saline in it does just as well, and in 5 days it finds the platform. But if we look at the animal that have the blood protein infused into it, we see that in 9 days of the experiment, they never learn. They don't really are able to navigate or remember or learn the way to escape that water pool. Okay, so it's enough to recapitulate aging within 2 weeks of this blood-brain barrier dysfunction that we're doing. So now what happens in the other way? Can we actually block that? And we ask that two different ways. We have those, we have the transgenic animals and we have the pharmacological model. And we asked whether we change that in middle age and now the brain ages in a protected environment, does that protect the aging process? And the answer was yes. But what I want to show you today is the surprise that we had there, a control group in which we said, let's let the animals age. And we're going to get to them when the damage is already there. And now we're going to reverse or restore blood-brain barrier dysfunction, and let's see what happened. And what happened there was the surprising part, that when we're looking at those animals, here is our group again that shows this pathological neural dysfunction. We're treating them with a drug, and within days, we start seeing restoration of neural signatures of young animals. What happens when they're learning? We can't just put them in a pool because they need to swim, and you need to swim. You know, when you're a very aged animal here at the end of your lifespan at 2 years of age, there are other functions. But we found two different tests that only focus on the brain function. One is a maze in which they need to remember where they were before and go to the other side. And the other one is object recognition. We give them some objects and they need to remember whether they've seen them before or not. Is it novel? And how much time do they spend with a novel object? And in both cases, old animals are not doing as well as young animals, but 2 weeks of a restored brain environment when we fix their blood-brain barrier, and we can see that those aged animals perform just as well as young animals. So this is probably the best news that I can give you about brain aging, because what that means is that there is some kind of inflammatory fog that sits on the brain that hinders, that just doesn't let the brain act as well as it does. But when we are dissipating this fog, the brain function is there, and the aged brain works just as well as the young brain. So it really tells us something very exciting about brain function during natural aging, and it made me think about this local cue. So in summary, I've shown you that vascular dysfunction is a novel mechanism that drives this inflammatory response in the subsequent neuropathology, and I've shown you that in aging, but it's also, where we were able to show that in traumatic brain injury and in Alzheimer's disease. So it's a common mechanism that was not well recognized before. It starts early in middle age or within an hour of injury, of traumatic brain injury, and it's associated with this individual variation, and there's surprising and promising reversal potential. And this comes on the backdrop of many, many, many different clinical trials that were completely failed in treating neurodegeneration and treating traumatic brain injury. And this, I think, offers some kind of a neural target to think about in neurology, because we can localize and quantify the blood-brain barrier in ways that I've shown you to identify the right patients at risk. And then if we can treat the blood-brain barrier dysfunction and let the brain operate in a different environment, we think that this would be extremely helpful.
Daniela, that's really exciting. We have to move on to the group discussion.
All right, just to point out that if you're interested in the backstory behind it, Scientific American in May this year.
Awesome, great, super exciting. I think in the interest of time, we should get all three of our speakers together and we can field a couple of questions for Daniela or for the group. And actually, Daniela, a question that came up related to your talk is, because of this interesting similarity between accumulated injury and aging in the brain, is all of aging accumulated injury, or is something else going on?
Oh, I think there's similarities in the mechanism, but there's also differences. It's not necessarily an injury itself. It's the dysfunction of the blood-brain barrier can come from very different mechanisms in aging as it does in injury. This is something that us and others is trying to look at now, but as part of the wear and tear of vascular systems that one sees in aging, in the same way that you would see that in the heart, for example, there's a gradual decrease in blood-brain barrier function that is not necessarily induced by injury.
All right, great, thank you. And actually, I have a question for maybe the whole group. You guys talked about your individual, research advances. What about your groups who do this work? You're here at Berkeley working with exciting students, and I'm wondering, how you think students, both undergrads and graduate students, contribute to work in your lab. How does it all fit together?
Okay, well, I'm happy to share. So, of course, these are the people who drive our research and the ones that actually do the work. The brain organoid project that I talked about was really the brainchild, for lack of a better word, of a very talented graduate student in the molecular and cell biology department. And he kind of developed this whole pipeline and was really incredible achievement. And just by reading, a paper about this emerging technology, he was able to establish this in our lab. So with a lot of help from, various rotation students and undergraduates along the way. So definitely they deserve all the credit.
And Michael, how do you bring together engineers and neuroscientists to do your work?
Yeah, I was on to just compliment also what Helen said, and I totally agree. One of the nice things I think about Berkeley is that kind of really complete integration, from the level of like undergraduates and graduates and postdocs, and the fact they all work very collaboratively together, both kind of at the different levels of education and training they receive, as well as Dan, as you mentioned, across departments, right? And so people can very easily here. We have so many programs at Berkeley which combine engineering and biology and physics and social sciences, where people can get to your lab through almost every avenue, depending on their scientific interest. And I think that's one of the strengths of kind of basic science approach, this ability to integrate vertically and horizontally across all levels, which basically allows you to go for a very creative type of question that would be difficult to do otherwise. So absolutely.
Great, well, thank you all. And at this point, I need to thank our speakers and turn it back over to Dean Botchin for some closing remarks. Mike, the floor is yours.
Thanks, Dan, and of course, thanks to Helen, Michael, and Daniela for sharing a very exciting glimpse into the frontiers of brain research. And I think it certainly has sparked the intellectual curiosity of the audience, and that's really what we're trying to do. There are just so many questions to follow up on the talks, and I'm always a little disappointed that there isn't enough time to get into a deeper conversation. But I encourage the audience to reach out if you'd like to learn more about anything that's been covered. We'll be sharing additional resources with you by email about today's topic and speakers, and a video of today's sessions will be sent with the URL for the site in approximately a week from now. And let me just advertise the next event. We're looking forward to seeing many of you at our next Basic Science Likes the Way event, which is going to be about extremes on May 4th. So until then, stay curious. Fiat Lux, and of course, go Bears.